Can people hear in the back?
You guys.
Hello? Okay, hi everyone. This is the bio285 exam review. I'm just gonna go over some announcements first before we start, since we're gonna start at 7. Okay, so first thing, your exam is tomorrow. Shouldn't surprise any of you since you're all here. You all knew that. And your exam, if you're taking it at 7 p.m., raise your hand if you're doing it at 7 tomorrow. Okay, that's like most of you. Okay, so that's in Mahar. And if you're doing it other times, there's like another place, I think French Hall, where you're taking the exam. And all the exams will have TAs as proctors, and Laura will be making her way around the rooms too to help answer questions. Big thing with your exam, please make sure you take your time. When I took this class, I was always the last person to finish my exams because you get like all the time you need to work on it. You'll notice in other classes, professors will try to like collect your exam after like an hour and a half or 2 hours on the dot, but Laura gives you all the time you need to do the exam, which is nice. Another thing I wanted to mention is please make sure for exam day tomorrow that you bring your cheat sheet. I had some students last exam forget their cheat sheet, made me sad because I didn't get a cheat sheet when I took this class. So the fact that you guys are allowed a cheat sheet, just make sure you bring that tomorrow. And it's a double-sided piece of paper, so you can use 2 sides. Someone emailed me about a typo in my slides. So for your cheat sheet, it's recommended but not required that it's handwritten. So in other words, we're not gonna go around and check your cheat sheet, and neither is Dr. Francis, but it would be in your best interest to write that by hand, in my opinion, just because when you write things by hand, your brain interprets information a lot better than you typing it. But that's just my opinion. And if you wanted to, you could bring your sheet from exam 1 with you to exam 2, although the exam's not cumulative, meaning that stuff on like trip operon and nuclear import export's not gonna be on this exam. Does that make sense for people? Okay, so your exam tomorrow, concepts that will be on it is pathways, microtubules, cell cycle, and that's what we're gonna talk about today. And some people I don't think knew at this point, but I hope everybody knows we have a YouTube channel. So we record all the review sessions we do and we post them on YouTube. So that's just extra information. And included in your week 6 review session slides, there's tips for how to approach this class. I took time to figure out some tips to give you guys on how to approach 285, which I got 2 years ago when I took this class from the TAs. So I thought I'd just pass it along to you guys to read about that. So hopefully you guys have read that info. It's on the week 6 slides, and I also said it verbally at the exam 1 review. Plus, if you got the slides from me today, you may have seen those. Another thing I'm gonna stress is please get a good night's rest for the exam. Please sleep, your mental health is important. You shouldn't be trying to pull all-nighter for this exam because you need to have your rest so that your brain can think of responses, okay? That's important. Make sure you eat dinner as well before the exam, or eat something. Could probably even bring a granola bar to the exam. Like I said before, you won't be tested on exam 1 information, but there's still concepts from the previous weeks that you should still be familiar about. So things like GEPs and GAPs, those proteins are important. And there's some examples of those in the pathway. So don't forget like what those do and what that means. And other concepts as well, like what it means for a protein to have higher or lower affinity for a protein or a ligand. Like for an example I wrote there, G alpha having low affinity for GDP. And then other things like what's considered to be an enzyme versus what's not an enzyme. Just understanding what enzymes are, what they do, that's like an overarching theme for this class. And one more thing I wanted to mention today is the slides in the recording will be sent to you, already sent the slides. And if there's any major changes in these slides that are different, please let me know, I'll send the new slides. But try to take in today's review and soak it in like a sponge. I don't want people to stress too much about trying to write down like all the notes and not absorbing what we're saying to you. Because I feel like what we verbally say to you is like more important than trying to write down all the notes, if that makes sense. I feel it's kind of similar in lecture as well, but you guys will see what I mean when we get to more of the review stuff. So does anybody have any questions on anything I just said so far? Yeah?
What would you recommend for us to write on the sheet?
Ooh,
Like
good.
problem-type questions
Good question. Okay, so I wonder if I'm gonna address this in my slides. No, so the question was, what do you recommend we write on your paper? I would definitely draw the pathways on your paper. You're not gonna be given the diagrams for this exam. There'll be no diagrams clipped to the back of your exam. So just have questions. So definitely drawing out the pathway would help. And I recommend doing that by hand. Although, like I said, handwritten cheat sheet is recommended but not required. So you do what you want with that information, okay? But yeah, pathways for sure. And I'll get back to you on that question actually, because during the review, you'll know what to write and I'll symbol it out. You have another question?
Yeah, but we are like allowed to bring a species, right?
So if you had a sheet from exam one, you could bring that with you if you wanted to. My understanding is that it's like the double-sided cheat sheet plus like the exam one sheet if you wanted to bring it, I believe. That's what she said. I could double check with her at the end of the review and text her and double check. Any other questions so far? Oh, got another question? Yeah. Yeah, I did. So I sent those out when? During class, I think. I have lab on Wednesday, so I sent it out earlier. But yeah, you guys have these slides already, but if there's a slight shift or change in these, because I added a little extra oomph to these slides, then I'll resend it. Okay, so I'm gonna go through these tips. I already showed you guys these already, but I think it's good to emphasize. How to approach this class 25, because this class is definitely different than most classes since it's not really memorization based exams where you regurgitate information from lecture. This is more problem solving techniques. So some tips and advice I put here: using your quizzes for review of just more than multiple choice, taking your multiple choice questions and trying to manipulate those into open response questions, or even just saying that like, oh, in this question, the answer is B, but why is the answer B? Like explaining the reasoning behind those questions and just practicing things like that of being able to explain your reasoning, I think is a good tool to use. Another thing I definitely think is great is group studying. If you don't have friends in the class, make them. You guys all hear the review session. I'm sure people have Snapchat, Discord, whatever. You guys can make group chats, study together, use each other as resources. Obviously, not during the exam, okay? But when you study together, you can. And why I think this is a good idea is because group work exposes gaps in your knowledge. When you're telling somebody how like the survival signaling pathway works, and if you make like a small mistake, the other person can say, no, but it works like this. But during that conversation, you're learning and you're picking up on your gaps in knowledge. So that's great, a great tool. Definitely using Laura's questions for sure, like I said, and engaging in something we call active studying, which I'm gonna talk about a little bit more. So basically the idea behind active studying is engaging with the material. It's engaging with the material in a productive, what we call productive way. Before I took this class, I had no idea what the heck active studying even meant. So I just thought I'd put some examples up there of what it means. So just like I said, doing practice problems or making up your own problems based on problem sets, quizzes, taking components of pathways and asking yourself or friends, what would happen if there was a mutation in this, or if we change something in this protein, XYZ, stuff like that. And usually in this class, like form follows function, which means if you change your protein shape or conformational, there's a conformational shift, then there's a change in protein behavior or function. So you'll see that theme across the semester. Drawing out the pathways, I think, is a good way to study because it reinforces your knowledge. You won't be required to memorize the pathways, but if you can like practice drawing them out just on a blank sheet of paper, it'd be a good way to like internalize the information. Rewriting notes by hand, I think, is a good tip for this class. As Laura said, it's scientifically proven that when you write things down by hand, you remember them better. It's just the way that your brain processes information. When I took this class, I rewrote my notes by hand, and I found that to be helpful. Another thing, big one, I think, start your problem set early, sooner rather than later. I think if you start it on like Sunday, the day of, or like Saturday, you're gonna be stressed out. And I just don't think that that would be best to do. So definitely start your problem set early if you can. I try to do like 1 to 2 problems a day and try to visit Laura and the TAs at their help sessions, which is like our office hours. Definitely doing stuff like that, I think is gonna help you in the long run. So if you guys have seen, the problem sets kind of simulate, the problem sets kind of simulate what the exams are like in terms of this critical thinking skills and think like a scientist thing that Laura is trying to teach you guys. So don't be afraid to reach out to Laura directly for help. She's very friendly compared to some of the other professors I had in my experience. I think I could say she's one of the friendliest professors I've met. TAs were also friendly as well. If you go to the review sessions, I call it in the flesh, but if you go in person to our review sessions, we do go through problem sets after we've covered weekly content. So if you appear in the flesh, live in person, we go through those things. So if you're there, you're able to get help with the problem sets. Those review sessions are Thursday, 7 p.m., right? Yeah. But yeah, using these TA review sessions is a great way to help you in this class. And yeah, a big thing also, I think, is understanding why you got a question wrong when looking back at like past exams or problem sets and explaining your reasoning for a particular question. I think that thing is vital, like learning about your mistakes and overcoming them, I think is a great thing for this class. So now I'm going to compare that to, so that's active studying. So passive studying is the opposite of that. So when somebody passively studies, it means that they only, or as I put in there, solely just reading books and watching videos and just not taking notes and not engaging with the material. And you're not really taking it all in. If you just like sit there and watch like a recorded lecture and you don't take notes, or you're like on your phone or something, you're not really absorbing much of what's going on because you're distracted and you're not really like there. You're there, but you're not there. I don't know if that makes sense, but seems people know what I'm talking about. But yeah, so the example I had was like listening to Echo 360 while you're on TikTok. You're not gonna, it's gonna be difficult to like pick up stuff. And then a big one, which makes me very sad and all the other TAs, is when students don't use the class resources when they need help. As you can see, I included a list of all the class resources. I'm not gonna read through all of those for time's sake, but all these things are there to help you. Use each other, use our help sessions, Laura's help sessions, review sessions. There's topic notes that Laura provides every week, very useful. And we have a TA in this class who sits in lecture every day and writes notes Monday, Wednesday, Friday. She posts those at the top of the Moodle page. So shout out to Emily for that. And another thing that would be like not the best thing for this class, for example, passive studying is not going to class because you're missing out on the iClicker discussions of the questions, which are pretty helpful. Any questions on anything I just said? Yeah?
You did the iClicker questions this week.
Would they be on the test?
Yeah.
From this week?
Yeah.
I can take a glance at what this week's were.
Okay, you can let us know
Yeah.
later. But I would go over clicker and probably problem set, I'd say. Okay, now another thing I want to bring up. I don't want to spook anybody because I want people to just focus on this exam and in this time period now. But I told some people at office hours and people looked at me like they were very shocked. But I'm gonna just mention this now, just so that you guys know, you guys don't have to stress out, okay? But there's something in this class called the final exam substitution policy. And what this means is that Laura essentially will replace if your final exam score beats one of your lowest midterm exam grades, she will substitute that grade in for your midterm. Now that might sound confusing to you, so I put an example here. So we have the student A here. They got an 80 on exam 1. Exam 2, they got a 78. On the final, they got a 75. Now this is an example where the substitution policy would not work here. Why? Because their final exam grade was lower than either of the 2 midterms. Okay, now we're gonna do student B. And these aren't like grading A, B system, it's just a random thing. So it could have been student X or Y. So now we have student B. Student B got a 73 on the first exam, an 80 on exam 2. On the final, they got an 85, which is great news for student B because they get redemption. So on the final, because they beat their lowest exam score, the 85 that they got in their final stays, and then their midterm 1 grade gets replaced by the final exam grade. So does that make sense for people, how that works? So it's not like an exam grade gets dropped, but you're just kind of like rewarded in a way if you showed that you learned the content to do well on the final, that you get rewarded with this like redemption thing. So if you did worse on the final, which usually is not the case, students tend to do better on the final than they think. But if you did worse on the final, this wouldn't happen to you. They wouldn't sub at the lower grade because that wouldn't make sense. Does that make sense for everybody, how that works? This is all in your syllabus as well, but I don't think people read the syllabus because at office hours, everybody looked at me like they were shocked. So I don't think people read it, but I thought I'd just mention it in here and say that one bad exam grade, however you define that for you, is not game over in this class. Some classes, when you do bad in the exam, sometimes you get screwed over and you're like, well, damn, like now what's the point of this class? And you get like really pissed off. But one exam grade is not the end of this class. You have a chance of redemption towards the end of the class. And I think that's a good thing to just mention. And this class is graded on a points-based system, which means that you have the 2 midterms and the final, which are each worth 200 possible points. But for the class overall, you can get up to 514 points. So there's plenty of other things you see as like problem sets and whatnot that all factor into your grade. You guys can read more about that in the syllabus because today we're going over the content. But does that make sense to people? Can I get a thumbs up? Thank you. Okay, now everybody is gonna take a deep breath from me, in and out, so that we are calm and we can move with the material. Ready? No, I'm serious because I feel like I can feel the stress in this room. It's so quiet. Okay, so can everybody do me a favor? Just inhale at the same time on 3. 1, 2, 3. And out. Excellent. Thank you. Appreciate that. Okay, we needed that. Now we're gonna go proceed. All right, we're gonna start with pathways. Sorry, I always like to have my footnotes. Okay, we're gonna start with pathways. You guys know there's 4 pathways for this class. I would, if I were you, I would draw them all on that cheat sheet of yours because they will not be offered as a photo on your exam. So draw them all, okay? And yeah, each pathway has their differences and similarities. So we're gonna look at each of them, okay? One thing I love to do, if you guys have ever seen my review sessions on YouTube or watched in person, I never do a review session without an analogy. And so I'm gonna do another analogy here. So basically, I want you to think of these pathways not as pathways, but as stories. And in a story, you have a sequence of events that occur in a natural order or in a chronological sense. And each story will have a different outcome and different characters. And there should be, theoretically speaking, what we call a happy ending, which is like the cell response in a pathway. And if you change a character's role or function in a story, then you may end up with a different ending. And basically, for your exam, you'll be presented with questions on pathways where there's possible mutations or changes to like a particular story. So you have to work around and figure out how to solve that. So in other words, there'll be changes in the original story, meaning that if you're able to understand a plot line or the original story or the normal pathway, you can make predictions as to what would happen if you change the script or flip the script. Does that make sense to people? Okay, and I'm actually gonna do some example of these problem solving questions today because we figured it'd be nice to do some practicing. And there's 4 stories, so let's just go ahead. Okay, first pathway, RTK, RAS, MAPK. Sometimes called growth factor signaling, but who cares? You don't need it to know that name. Okay, so how this pathway works, or how it starts, excuse me, is you have the signals in the form of a dimer, binds to the receptor. When the signal binds to the receptor, the 2 RTK monomers will dimerize, which is just a fancy way of saying they come together. And when the 2 receptors dimerize, that causes a conformational shift in the RTKs that causes the intracellular kinase domains to become active, which is the bottom, the intracellular portion of the RTK. And then once the kinase domains become active, they're able to cross-phosphorylate each other, or in other words, autophosphorylation. And then once the two RTK monomers cross-phosphorylate each other, we have the active RTK. Does that make sense? Does anybody have any questions on anything I just said? Can I get a thumbs up, please? Thank you. I rely on that so I can continue. Otherwise, I feel like people are confused and I can't tell on what. Thanks. Okay, so now at this point, we have active RTK, which is pictured here. So this is just the same thing, but a continuation. So active RTK is pictured here. And what will happen next is that you have the adapter protein, which I think Laura also refers to as GRAB2, but either name is acceptable. They mean the same thing. So adapter protein, which is pictured here, the dark blue protein, binds to the active RTK. And when that happens, that will cause the adapter protein to recruit the RAS activating protein. This RAS activating protein, the lighter blue, is also RAS-GEF. That's another name for it. But you can just stick with the diagram name. So adapter protein, so active RTK, because the RTK becomes active, and then it will bind to the adapter protein here. And then the adapter protein will recruit the RAS activating protein. And when that happens, the RAS activating protein will encourage RAS to exchange GDP for GTP. So RAS activating protein at this stage is helping RAS swap out GDP for GTP. And so RAS will drop GDP and GTP will float into the binding pocket of RAS. And when RAS is bound to GTP, it is now active. And in this diagram, RAS is anchored to the cell membrane in this pathway. Any questions on what I said so far at this point? Yeah.
So what's the benefit of having a lipid anchor? I just remember that from the comments that all I know is that lipid is
For this particular pathway, I'm not sure the benefit. Any ideas?
in general, just if you have lipid anchors, the idea that Professor Francis talks about is that because certain like down signaling things that needs to be activated, some of them are more concentrated near the cell membrane. And so if your pathway itself is like anchored to the lipid, then it's closer to whatever downstream proteins need to be activated next. So it's more like convenience and efficiency for the cell.
Yeah, I think it just probably makes signaling better because if RAS was freely floating in the cytosol, then it's going to take longer for it to find MAP kinase, which is the next step. So like having it anchored in the membrane, I think just helps signaling to be more efficient. Yeah, thank you, Tina. Any more questions on this part? And by the way, I broke it down into like little segments, but then there's a big picture of it all at the end. So I feel like it's too scary to look at it all at once before it's been broken down. Any other questions out there? No? Okay, I'm going to continue. So now at this point, RAS protein has become active. How do we know that? It's bound to GTP. So the next part. So now we have active RAS, and RAS, what it does is initiate what we call a kinase cascade. What is a kinase cascade? So basically, RAS will interact with MAP kinase. It's quite a mouthful to say. So I just say RAS will interact with MAPK3. It's important to say that RAS would interact with MAPK3. RAS is not a kinase, so it does not phosphorylate MAPK3. So notice how I said RAS interacts with MAPK3. So RAS will interact with MAPK3, and that causes MAPK3 to become active, which is the green one here. And then MAPK3, because it's a kinase, will phosphorylate MAPK2, and then MAPK2 will phosphorylate MAPK, or I think I said MAPK1. And then once this happens, MAP kinase, once it becomes active through the phosphorylation of MAPK2, MAPK1 will end up phosphorylating other proteins, which will lead to changes in protein activity, or in this pathway, I think, which is more emphasized, it'll go on to activate transcription factors that lead to changes in gene expression and result in cell division, which is our happy ending of this pathway, the cell outcome, cell division. So yeah, so that's basically the RTK RAS MAPK pathway. And at the end of the pathway, at some point, RAS will be deactivated because RAS is bound to GTP, it's active. So once it's done with signaling, it'll be deactivated by the help of GAP. If you recall from when we talked about nuclear import and export, what GAP does is it will bind to RAS, and it will accelerate the hydrolysis of GTP. So it turns GTP into GDP. Any questions on that? Oh, and RAS is a GTPase. So RAS is an enzyme. So it's able to intrinsically hydrolyze GTP into GDP itself, like RAN in the nuclear import pathway, if you guys remember. But GAP helps it to do it at an accelerated rate. Okay, so GAP is not the one hydrolyzing GTP into GDP. It just makes RAS do it faster. Does that make sense? It's important to distinguish that. Okay. Okay, so now I've explained to you the pathway, small little steps, but that's the entire pathway. I definitely would draw that pathway on your cheat sheet. So because it's a story, you have all the different characters in there, and the happy ending, which is the cell outcome, is cell division. So now I'm gonna actually ask one of you to tell me what would be a way that we could stop the happy ending from happening. How can I stop cell division? What would be something I could do to this pathway?
You could change the tyrosine
Yeah, I'd say that's a good idea. If you possibly change the mechanism in the RTK so that you said what you said, it was unable to cross-phosphorylate each other. Is that what you said?
Yeah, so basically cross-phosphorylate and the adaptive protein was able to bind.
Excellent. So you notice how she explained that. So if the RTKs were not able to cross-phosphorylate each other, they'll never become active. No active RTK, no adaptive protein binding to the RTK. No adaptive protein binding, no RAS activating protein. No RAS activating protein, or no, RAS activating protein not being active, excuse me, it will not activate RAS. RAS will be bound to GDP, no kinase cascade, no cell division. So that's just a way of explaining how altering one thing can affect the pathway. So that's just like an idea of that. Okay, so now I'm gonna ask you guys a question because we're all about active learning in this class. So what would happen to this pathway if RAS was always bound to GTP and it could never be shut down? Need somebody to answer this because you guys are gonna do this kind of critical thinking tomorrow. Okay, you're in the back, you raise your hand first. Okay, cell division will always keep happening and what? I missed that last little part. Okay, so I see most of what you said was pretty good. Only thing is, maybe because I feel like my question didn't say it, but the only thing I'm changing in there, the only thing in this pathway I'm changing is RAS. So GAP would still be there. It just wouldn't be able to do its job properly because maybe there's a mutation in RAS that's causing it to be stuck to GTP. Maybe it has a higher affinity for GTP, or it's just always constantly bound to GTP. But notice how she explained that because that was really good. So basically, if RAS was stuck to GTP, RAS will always be active. What happens when RAS is active? It activates MAPK3. Then MAPK3 activates MAPK2 and blah, blah, blah. Then you'll always have cell division happening and happening and happening. So this pathway would become hyperactive. And so your happy ending would just continuously happen you'd get massive loads of cell division. And that's the sort of thing that happens with cancer. So yeah, and so basically because I only changed one protein in there, which was RAS, GAP would still be there, but it would just struggle to hydrolyze GTP to GDP because RAS is like permanently active, so it'd be stuck to GTP. So does that make sense? The way she thought it through? Great job, by the way. Yeah?
I'm just a little confused. You think cell division?
Yeah?
I'm just confused where that comes in. So I thought we were talking about like if it's like geographic cell division, is that what you mean by that?
Okay, so when I say cell division, essentially what I'm saying is like each of these pathways, the purpose of the entire pathway is to take an external signal, transmit it inside the cell, and cause cell response to happen. And so the ultimate goal of this pathway, or the end game, is cell division. And that's not really pictured here, and you don't have to know like the details of it, but just know that MAP kinase, what it will do is it ends up activating a bunch of transcription factors, and then transcription factors, bind to DNA, cause transcription, and eventually down the line, gene regulation, expression, and that all of those events eventually lead to cell division. So that's what I mean by happy ending. And I think sometimes it's hard to tell with the pathways. I think people get caught up in trying to memorize the pathway and not think about the big picture of what's happening. But the ultimate end goal of all the events of the pathway, the big ending, is cell division. So by shutting down the pathway, you're stopping cell division from happening. Does that make sense?
Yeah.
Yeah. And it's a good question. Thank you. Any more questions? No? Can people give me a thumbs up? Okay. Sorry, just checking my speakers notes that I said everything. Yes, I did. Okay. And I was gonna ask another question. No, I think I'll ask it later. Okay, anyways. Okay, and so in these slides, I included a nice handy dandy summary of the pathway. Nice, very detailed stuff. I won't repeat everything, but it's there for you guys to see. So I'm gonna go through the next pathway, which is survival pathway. Let's see. So basically, purpose of the survival pathway is to promote cell survival. And in a cell, the cell's default setting is cell death. So what I mean by default setting, it's kind of like on your phone when you have like the language set to English. Like the cell's default program mechanism, if this pathway is inactive, is to cell death. So this causes the cell to die. So the idea is by signal binding to the receptor on this pathway and the pathway causing cell response to happen, that cell response is cell survival. So that's why this pathway is important. And if you think about it in a bigger picture context, this pathway is important for cancer research, because if you know, cancer cells are very harmful and we don't want them to keep dividing. So to stop them from dividing and surviving, we want to effectively try to shut down this pathway. So that's what researchers try to do with this pathway. So cancer is essentially hyperactive pathway. There's too much cell signaling occurring and growth of cells, which is tumors and all that. Does that make sense so far? Okay, stop me if you have any questions. Now I'm gonna just go through how the pathway works, just like before. So you have the signal binds to the receptor, RTK dimerization, and phosphorylation. Are you good with that so far? Okay, so then you have active RTK. And when RTK is active, it will have this protein here, PI3 kinase will bind to the active RTK. And once that occurs, PI3 kinase will phosphorylate an inositol phospholipid, which I'm pretty sure you can abbreviate as just IP. So yeah, so activated PI3 kinase will phosphorylate an inositol phospholipid. And before that phosphorylation event happens, that specialized phospholipid, not a protein, by the way, it is in a diphosphorylated state, meaning it has two phosphate groups. So when PI3 kinase reaches out and phosphorylates it, it makes it triphosphorylated. But because it normally is bound to two phosphates, we just call this new one, phosphorylated inositol phospholipid. So the action of PI3 kinase is pretty important because that's how we get PIP3, which is this phosphorylated inositol phospholipid. And once that occurs, the cell can use this in the rest of the pathway for binding to AKT and PK1. And basically, the idea is you want to get PIP3 because PIP2 is useless, essentially. So that's why we need PIP3 in this pathway. And like I said, it's not a protein, it's a lipid. And something interesting just to note is PIP3, the action of PI3 kinase, can be reversed by a phosphatase called P10. I'm not sure if she addressed this in the lecture videos, but P10 essentially is a phosphatase. And if you remember, phosphatases remove phosphate groups. So P10 is a phosphatase, and it can remove the phosphate group from PIP3, which would just make it regular inositol phospholipid, which is useless to this pathway. But essentially, you can imagine that if you remove the phosphate from PIP3, what the consequences will be. And you'll see that once I explain the rest of the pathway. Does this make sense so far? Do we have any questions? No? Okay. Okay, so once we have PIP3, we're good at this point. So the next part of this pathway is AKT will bind to PIP3. And something which I think she mentioned in lecture is because this is a cell, you'll have multiple molecules, of PIP3. Even though in your pathway there's only one pictured, imagine the cell has multiple molecules of PIP3, okay? When you're looking at this pathway. So AKT will bind to PIP3, AKT is this green protein, and also protein kinase 1 will bind to PIP3. So at this point, this happens, and then probably arguably the most important part of this pathway is we need to make AKT active. So to make this happen, you have PIP3 binds to AKT here, and then you have to have the phosphorylation of AKT has to happen twice by two different kinases. So protein kinase 1 will phosphorylate AKT on this side, and then the phosphorylation of AKT by PK1 causes a conformational shift in AKT, which allows it to expose amino acids on its other side so that protein kinase 2 can phosphorylate it on the other side. Anybody have any questions on that so far? No? Okay, and so this event of PIP3 binding to AKT and protein kinase 1 and protein kinase 2 phosphorylating AKT, all those together causes AKT to become active. And once AKT is active, it'll dissociate from everything and float around in the cytosol. Does that part make sense so far? Got a question? Mm-hmm. The order of it, I think she said like it didn't matter. Like one of the lectures, I think she said like the order of that doesn't matter. But just know that like for AKT to become active, those three events need to happen. So you have PIP3 sits in that binding pocket, that's a pH domain. But I'm going to talk about domains afterwards, I promise. And then phosphorylation of protein kinase 1 has to happen first before the phosphorylation of protein kinase 2, because there's two spots on AKT that need to be phosphorylated for it to be active. Does that make sense? So those three events together, we have active AKT. Is that good? Any more questions? These are all good questions, by the way. Good clarifying questions. Yep. I'm gonna say yes. I believe so, because it has that binding pocket for it. So I'd assume so. So anything that's so like if it was not there, the PIP3, you can make predictions about that, but I don't think that'll be on. I think so. Yeah. I'm gonna go over domains. I know I just said pH domains. People are like, what is that? But I promise I'm gonna go through the domains. But the pathways have domains, and those are just like binding sites for proteins. But I do have a slide on that. But I will go through that. Is that a question? Yeah. Okay. So I trying to figure out what you mean by that, but basically, an inositol phospholipid is anchored to the cell membrane. So AKT will bind to PIP3, which PIP3 is anchored to the cell membrane, and then it looks like because PIP3 is on the cell membrane, protein kinase A will also bind there. So AKT has to kind of position itself at the top to be able to bind to PIP3. And then because protein kinase 1 is also bind to PIP3, it's kind of like restricted to like that area. I guess like Tina, I don't know if you want to add, but I remember her
Are you
saying
trying
like
to-
the order
Are
of
you
that
trying to
doesn't
ask if
Let's
AKT is already there or?
I don't think she actually knows that, like Dr. Francis. But I wouldn't get too tripped up on that. Just I don't think that's like the most important part of it. I promise you. Okay. But basically, when you have PIP3, it'll sit in protein kinase 1, and then another molecule, PIP3, will sit in AKT. And so for AKT to become active, like I said before, 3 events have to happen. You have your PIP3 sits in AKT at that domain there, and then you have to have the phosphorylation of both protein kinase 1 and protein kinase 2. But I do remember her saying that protein kinase 1 has to phosphorylate first so that it exposes the amino acids on the surface of the other side of AKT so that protein kinase 2 could come in there and phosphorylate it. I remember that part being important, 1 and then 2. But in terms of whether PIP3 goes first or protein kinase 1 goes first, that's not something to get tripped up on, and I don't think you'll be tested on that. Okay? Any other questions? I'm happy to answer any questions. It's good to get the questions out there before tomorrow. Is that a question in there? Okay, we're just stretching. Okay, cool. Anyways, question. Yeah. That's correct. Well, there are 2 protein kinases pictured here, but you mean them both?
So protein 1 does it first.
Excellent. Couldn't have said it better myself. Okay. So now at this point we have active AKT. And by the way, these are great questions. Okay. So active AKT. When AKT is active, what does it do? Swims around in the cytosol. So you see it swimming there. Anyways, so now at this point, AKT is active and it will phosphorylate BAD. And BAD is the blue protein over here. So once AKT phosphorylates BAD, this part is key, okay? Once AKT phosphorylates BAD, BAD becomes inactive. This part trips up students a lot. I know it can be tricky to understand because most of the time you see phosphorylation activate stuff, but in this pathway, when AKT phosphorylates BAD, it makes it inactive. Okay. And so when BAD is inactive, BAD will actually release BCL2 and bind to some other protein over there. Nobody cares about that gray protein over there, but just know that when BAD gets phosphorylated by AKT, this action here causes BAD to let go of this red protein here, which is called BCL2. But yeah, so before BAD is phosphorylated, let me start there. Before BAD is phosphorylated, it is holding this red protein prisoner. This red protein is BCL2. But when BAD gets phosphorylated, it becomes inactive and it releases BCL2. So BAD at this point, is active when it's holding this protein prisoner. But when AKT comes along and phosphorylates it, it becomes inactive and it's bound to this protein, but no one cares about that. Just know that when BAD is inactive, it's not bound to BCL2. I'd say that's the more important thing. And so, the part of this pathway that's important is the happy ending. So basically, when BAD is phosphorylated, it becomes inactive. Then what happens next? Then BCL2 breaks free and saves the day. How does it save the day? It stops cell death from happening. Inhibition of cell death, in other words, cell survival. And so that's how that pathway ends. The cell survives. Yay. So that's that pathway. Any questions so far? Because I know that part can be a little bit tricky to understand. No questions? You guys had like 8 questions for me earlier. I love the questions. No? Okay, and does that make sense? Can I get a thumbs up? Excellent. Okay, I was gonna guess that it didn't make sense, so I have more stuff here. Anyways, no, because this part can be confusing. That's why I always put extra slides on it. Anyways, so what is apoptosis? Cell death. BAD is a protein that initiates cell death, which means causes it to happen. And then BCL2 is a protein that inhibits cell death or apoptosis. There's two scenarios that could possibly happen. You have active BAD, and when BAD is active, it's bound to BCL2. And then BCL2 is not active when it's bound to BAD. So then you get mitochondrial holes and cell death, sad face. Then you have the alternative, which is when the cell survival pathway is working properly. Active AKT will phosphorylate BAD. Then at that point, BAD becomes inactive. When BAD's inactive, it's no longer bound to BCL2. BCL2 is active and it's free to save the day, which means that it'll bind to mitochondria, stop cell apoptosis, so the cell lives. Smiley face. Okay, I have an interesting analogy here that I thought I'd explain. How many of you have seen Phineas and Ferb? That's most of you. Okay. In case people had never seen it, I was actually going to include a link actually. But anyways, if you guys have seen the show, you know that Dr. Doofenshmirtz, this guy here, is evil. He's bad, okay? That's why I made this analogy. And so when Dr. Doofenshmirtz is doing bad things, the whole city's in trouble. It's not good. And he's a bad character. But Perry the Platypus always comes in and saves the day. So I try to think of that in the context of cell survival. Because when that is active, you get cell death. But if BAD is inactive, when it's phosphorylated, then BCL2 can like break free and save the day. And in the TV show as well, Dr. Doofenshmirtz always traps Perry the Platypus, but then he breaks free and he saves the day. And then Dr. Doofenshmirtz, he doesn't die. It's not like cell survival, but he just like, ends up, his own invention ends up shooting him in the foot or something. Whatever. But anyways, just an analogy I put in there. But yeah, so basically BCL2 is held in captivity by BAD protein. Same thing as a TV show. Okay. And then if BAD becomes inactive, then BCL2 can break free and save the day and cause cell survival to occur. Just my analogy. I don't know if you guys like it or not, but anyways, I was really proud of that. But anyways, so basically main idea is if you inhibit the inhibitor of cell death, the cells will die. So that's like a double negative, but basically what I'm saying is if you inhibit the cell survival superhero, which I think of like as BCL2, the cells will die. That's scenario one, which I just said. Scenario two, if you actually phosphorylate BAD, and then it releases BCL2, and it can save the day and cause cell survival to occur. Does that make sense? Any questions on anything? Do people like the analogy or no? Thank you. Appreciate it. Anyways, now we're gonna ask questions. So the happy ending of this entire pathway or story is cell survival. So I guess my question to you would be, how could I cause this cell to die? Can anyone think of any mutation or anything we could change about this pathway to cause cells to die? I just need one volunteer. Oh, by the way, I have candy. So if you volunteer to answer my questions, you get candy. I was gonna give people $1, but no, I'm not rich like that. Anyways, I think you answered already. Actually, if you answered already, come collect your candy. But let's see. Okay, go ahead. Give me an example.
If you prevent the signal from binding to the tyrosine kinase, then the entire cascade effect can occur, and then BAD will be phosphorylated, and then BCL2 will be sequestered, and then the cell will.
Wait. Say it again. Wait, repeat yourself. I'm sorry. I think I missed part of it.
That's okay. So I said if you prevent this route signal from binding
Okay.
to the tyrosine kinase,
Okay.
the whole cascade effect won't occur. AKT will be phosphorylated, and then therefore BAD won't be phosphorylated, and that means BCL2 will be sequestered by BAD, and then the cell will die.
Excellent. Okay, because I thought you said BAD would be phosphorylated at first, and I was like, wait, no, I'm just correcting you. That's all, because I'm annoying like that. But anyways, what he said was a great idea. Take out the signal. Nothing would happen. Obviously, you have to, put in detail what does nothing mean. So you just write nothing will happen. That's a little vague. But if you take out the signal, no RTK dimerization, no cross-phosphorylation, no active RTK, no PI3 kinase activation. then you'd be stuck with PIP2. If you're stuck with PIP2, then there'll be no way to activate AKT, and no active AKT, then what part are we at now? No active AKT, then you're welcome. Then I lost my train of thought. Then that wouldn't be phosphorylated, and then cell death. Boo-hoo. Okay. Any questions? No. Okay.
Did you emphasize P10?
Yes, I did. I emphasized P10. Okay. But yes, I emphasized P10 earlier. I would write that down on the cheat sheet. P10, ladies and gentlemen. You heard it here first. Okay. This is recorded, by the way, so you can go back and see. But anyways, I'll show you because I'm nice like that. Anyways, P10, I said earlier. Where the hell is this? Excuse me. Anyways, P10 down here. It's not pictured in your pathway. A P10 is a phosphatase that has the ability to remove phosphates from PIP3, which makes it PIP2, which if you recall, I said PIP2 is useless to us in this pathway because we need PIP3 to sit in AKT and molecules of PIP3 to sit in protein kinase 1. So I definitely would write that down. Hint, hint. Okay, question in the back? Oh, a question using P10. Ooh. Okay. Wait, hang on. Wait. Where's this thing? Wait, let me. Hang on, give me a second here.
Do you want to take a stab at that? Like you said,
Oh,
if
yeah,
P10
that's
has a
actually good.
gain of function. So if P10's always doing its thing, what would happen? What do you think?
Okay, but
But
why?
why? Yeah.
Let's say this was a question hypothetically. Explain your reasoning. Okay, you're right. If P10 was active, you'd get cell death. But why? Very good. Okay, but I know that you know. From that, I could tell she knew. She understood the assignment. Okay, full credit. Okay, yes, question.
What if AKT was already
Ooh, I don't think she's gonna try and trip you up like that. I don't think. But so just take the order of the way I have it in the slides. Just roll with that. Okay, just trust. Okay, so when you have PIP3, it looks like a mess in this part, but I like the way I made it better. Where is this? So when you have PIP3, okay, the next part, what does it do? It sits in protein kinase 1, and then another molecule, PIP3, sits in AKT. So you can assume at this point, sorry, I'm blanking out on the name. Who asked the question? Sorry, I was just saying for PIP3, you said is AKT active or not? But remember, I said 3 events need to happen for AKT to become active. So you can have if PIP3 was here, but if PIP3 is here, it wouldn't matter because you still need protein kinase 1 and 2 to be there for activated AKT. I emphasize 3 steps. But if you had P10 thrown in here and it was always active, P10 would remove a phosphate from PIP3, so you wouldn't even have PIP3. You'd be missing it. You'd have PIP2, which is useless. And even if you had protein kinase 1 and 2, AKT wouldn't be able to become active. Why? Because we need PIP3 also as well.
I think unless she specifically specifies in like a question that AKT is active, you can assume that it's not yet active.
Great, what Tina said. Tina, say it one more time.
What?
Say it again
Unless
for the video.
Unless Professor Francis specifically specifies that AKT is active, then you can assume that like whatever she's asking, it's not yet active. And that's how you should think about the question.
Okay, excellent. So that's survival pathway. That's 2 of 4. We're getting there, people. Do we have questions? Do you have questions? No? Okay, I'm gonna try and go through these because we still got cell cycle and that stuff.
This is like the brunt.
But this, I would say majority, because you got 4 pathways. And I think 7 questions. So I think, let me see here. I'd say 4 of your 7. But don't panic. You guys got this, okay? What?
We don't know specifically, but like Laura said in class 2, the pathways are.
She said,
majority.
okay, she said more of pathways will appear because there's 4 of them. And then she said more cell cycle as opposed to microtubules. That's what she said in class. I'm quoting what she said in class, guys. Okay, okay, please, please. I want you guys to be out of here by 9 p.m. hopefully, so you guys can get a good night's rest. So I gotta continue. So can I have quiet in the house, please? Hello? Excuse me? I need to be quiet. Sorry, I hate doing that. Anyways, next pathway is JAK-STAT pathway. I need to be quiet in here, please, ladies and gentlemen. Thank you. With respect to the YouTubers who are gonna watch this later. Anyways, so JAK-STAT pathway. How does the pathway work? Can you explain it? And big central idea is, would the receptor used in this pathway be an example of an RTK? Actually, I'm gonna ask you guys, is the receptor in the JAK-STAT pathway an example of an RTK? No, I'll give you all candy for that. But why? Anyone want to try to take a stab at why? If you've seen the slides, you probably know the answer already. But why? Anyone? No? Okay, you're close. Anyways, okay, I'll give you candy for that. That's acceptable. Okay. Anyways, so JAK-STAT pathway. Oh, I forgot to say. End outcome of this pathway, the happy ending here is you transcribe genes that are needed for breast milk production. So this pathway makes milk proteins for breast milk. And as a result, you get a happy baby. Yay. Okay, we got a question already? Okay, no, it's not a bad thing. When you say only, I wouldn't say it's only because there's like multiple different outcomes for different pathways. But for the focus of this class, 285, tomorrow's exam, that should be the outcome of the pathway. Unless she says otherwise, unless she talks about a question where it creates purple proteins for aliens or something. But no, I don't think she do that. That's okay. Any other questions? But that's the happy ending of the pathway, okay? And receptor in this pathway, I want everybody to be quiet, please, because this is key. And as your TA, I care about you, try to help you here. Receptor in this pathway is not an example of RTK. Why? Because the green receptors in this pathway, the prolactin receptors, they don't have kinase activity or kinase domain. These are just plain old simple receptors. And essentially what's going on with this pathway here is the receptors themselves, which is the green parts, do not have kinase activity, but it's the JAK proteins they're attached to, which JAK stands for just another kinase, by the way, that's his name. The JAK proteins have kinase activity, and they're the ones that do the cross-phosphorylating. So he was right. Fantastic. Okay, and you have a question.
Do it again.
Yeah, I wish I could repeat it perfectly and just rewind in real life. But okay, let me try it again. What did I say? It was too good and I forgot. Anyways, so anyone else want to? Anyways, so the receptors used in this pathway are not an example of RTK. Why? Because receptor tyrosine kinases, their proteins and built into their receptors is an intracellular kinase domain portion that's able to cross-phosphorylate within the receptor. But these receptors are different. So it's not the actual receptors themselves, these green things, doing the phosphorylation, doing the cross-phosphorylating. It's the JAK proteins themselves. I mentioned this in the slides. So yeah, the JAK proteins themselves do the cross-phosphorylation, and that is what causes them to become active. Then they activate the receptor. So that is key. Okay? And I said it like 10 times in this slide. I repeat, it's not an RTK because they don't have autophosphorylation mechanism. And JAK, if that helps you to study for it, JAK stands for just another kinase. Okay, does that make sense? Why it's not an RTK? Because she said it before, I'll say it again, not an RTK. Okay, write that. Okay, next, how does this pathway work? Signal binds to the receptor. Prolactin signal, which is like milk hormone, I think. Thank you. Signal binds to the receptor. Receptor dimerizes. But the JAKs cross-phosphorylate each other, the blue proteins. Then the activated JAKs, like I said, not cross-phosphorylate, excuse me. The activated JAK proteins phosphorylate the receptor. Then at this point, once the receptor has been phosphorylated, we have these peachy proteins here, STAT5. They will bind to the receptor here. on either side of these receptors. And once they bind to the receptor, they're in close enough proximity to the JAK proteins that the STAT5s can be phosphorylated. And once they become phosphorylated, these STAT5 proteins, once they become phosphorylated by the JAK protein, they will dissociate from the receptor. And when they dissociate from the receptor, they will cluster up together to form what we call a dimer, an active STAT5 dimer. Okay, and then when they form a dimer, because STAT5, they're like a, I don't know what you call that, like a double agent or what do you, like a, somebody that has like two roles or whatever. I don't know what you call that. I don't know why I'm thinking double agent, whatever. Anyways, I'll come back to my thought later. But like they have like a dual role. So they act as like protein and transcription factor because they can enter the nucleus. And because they're transcription factors, they bind to DNA and cause transcription to happen. And then you have your big happy ending, which is the transcription of milk protein genes. And that's it for the JAK-STAT pathway. And how do they get into the nucleus, by the way? Just a fun fact, important. Basically, if you remember important from the last exam, important brings stuff to the nucleus. But yeah, that's your pathway. Any questions? No question? Okay, we're good. Okay, we're going forward. All right, you know what I'm gonna ask you. So we have happy ending, breast milk protein production. In other words, happy baby. How can I make the baby sad? How can I cause no milk protein production? Somebody give me an example. I have so much candy here. I'm just gonna give candy to everybody at the end. It's fine. Example? Okay. You wanna give an example though? Get extra candy? Yeah, go ahead. Give it a shot in the back. Good, that's a good idea. So if there was a loss of function in the JAKs, maybe their like kinase activity causes them not to be able to cross-phosphorylate each other, then that would mean we'd be here, right? So no cross-phosphorylation of JAK protein, and then that would mean the JAK proteins would not be able to phosphorylate the receptor. No phosphorylation of the receptor, no STAT5 no STAT5 monomers binding to the receptor. And if the STAT5 can't bind there, they won't be phosphorylated by JAK proteins. So no STAT dimer, no transcription factor sneaking into the nucleus, and no milk protein production. It's sad baby. Okay, good example. Candy for you, extra. Okay. Question.
Oh, I just have
Example.
yeah.
Oh, go ahead.
A mutation in SH2 domain.
Mutation in the SH2 domain of STAT5, causing it to not bind to the phosphotyrosine binding site on the receptor. That's a good one. Candy for you. Okay, great. We're going to talk about domains next, actually. That reminds me. But yeah, any questions? You got a question in the back. Ooh, good question. Okay, you guys, this is active learning. You guys gotta come up with these ideas. Good practice. Okay, go ahead.
If JAKs were permanently phosphorylated, like there was no way to phosphorylate the JAKs.
Okay. Okay, so if the JAKs had like a gain of function, okay? So even with or without the prolactin signal, if the JAK proteins were always cross-phosphorylating each other, then you'd have the active said that wrong. So if the JAK proteins were always cross-phosphorylating each other, then they can phosphorylate the receptor. Then the STAT5 binds to the receptor, and then they get phosphorylated by the JAKs because they have gain of function in their kinase activity. Then dimers, and then dimers will enter the nucleus, and then you get overproduction of milk, so too much milk, which doesn't sound that bad, but like hyperactivation of a pathway, too much like cell response is a bad thing. You want normal cell response. But yeah, actually, there's a video in this slideshow that shows that. So if you want to watch that, go ahead. Any questions? Oh, sorry, I couldn't even see it right in front of me. Yeah.
So the receptors still have tyrosine, but they're not RTK.
Wait. Mm,
Because I remember one of the questions was like turn the tyrosine into phenylalanine and the
No,
potassium
but that
to-
was with RTK because RTKs, I have this in my slides, but RTKs are receptor tyrosine domains.
Right.
And the intracellular portion of the RTK is composed of kinase domain. And when you phosphorylate the, what's my example? When you phosphorylate kinase domain, when you phosphorylate the kinase domain, it will cause this like, it'll cause a binding site, the SH2 domain here on the phosphotyrosine domain. But that's with RTKs.
Right.
And Jack is not an RTK. No, 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 please. On the exam, no, 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 it's not.
But so then what's the change to the test? If I forget, I remember.
Okay.
Something like if the JAKs can still phosphorylate because they're not part of the receptor, they're like subunits. Something about.
Yeah, so I guess for that question, I guess if the receptor was, if the signal bind and the receptor dimerizes, if the JAKs are in close enough proximity to each other, they will be able to cross phosphorylate each other. Because if you remember the sequence of events, signal binds, the receptors come together like this, and then because the proteins are in close enough proximity, they're actually able to cross phosphorylate each other. So that's the idea behind that. But yeah, which is different from RTK. Notice I said it like 7 or 8 times. So it must be important, right? Must be. Okay. So, oh, another question. I love it. Okay. We're gonna get to the cell cycle, I promise. Yeah?
Can you just remind me what
Receptor dimerization. Dimerization is just a fancy way of saying they come together. So signal binds, usually this the sequence of events. Signal will bind to the receptor, causes the receptor to dimerize, which is come close in proximity. And in the RTK, when the receptor goes like this, they cross phosphorylate each other because it has a kinase activity. Excellent question. Any other questions? You got one more pathway to go, and I promise you my time to talk will be over, I promise. Okay, wait, before we do that, I thank God I reminded myself. Domains. Okay, so most of the pathways we looked at have domains. What are these? Okay, so domains are binding sites for proteins. They have specific functions. For the purpose of this exam, you'll have to know how to spot domains on the pathways and ideas behind them. The 3 main domains we talked about in this class. The main one, or not main, but like the most common one, is the SH2 domain. SH2 domain, what is it? A phosphotyrosine binding domain, or area on phosphorylated tyrosine of a protein that a different protein can bind to. Most commonly used in our pathway. Plenty of examples. An example of this would be binding site on active RTK, which allows it to bind to the adapter protein. So, oh geez, I don't know if anybody wanted to see a picture of this. But yeah, so example of SH2 would be here, okay? SH2 domain on the active RTK binding to adapter protein. Does that make sense? Can I get a thumbs up? Okay, great. Ooh, gotta find this. Okay, so that's SH2, most common on most pathways. Jack Stack, can anybody give me an example of SH2 domain? Okay, but where? Because I see stat in 3 different places in this image. So where are you talking about? On the receptor, the stats themselves, or what are we talking about? Stats themselves, that's right. So the stats themselves, they will form a dimer with each other by binding to the SH2 domains. Ooh, that's a good one. Look out for that one. Okay. Okay. Anyways, and in your summary of these pathways, which I'm skipping over because I explained it all, we have examples of domains because your TAs are so nice. They wrote the examples of the domains in there too. So, you know, anyways, next domain, pH domain. We only have examples of this one in the survival pathway. Okay, so survival pathway is the only one for this class. Where is it? Oh, that has examples of this. And this is basically, sorry, I need this. And this is basically a binding site for an inositol phospholipid when it's in the triphosphorylated form for PIP3. So could anybody give me an example, or just shout it out real quick, of where there's a pH domain in this diagram? That's right, PK1, okay. AKT, excellent. Okay, you're getting the hang of this. Fantastic. Okay, a question, yeah.
We go back to step five.
Yeah.
So with regards to SH2 domain, if there's just scientific cholesterol like a paraffin or protein, would there also be an SH2 domain like I mean obviously there has to be one of the top set that's being used during that binding for the center.
There is, okay, so when I say between two proteins, could also be used for receptor as well, because RTK is an example of a protein, but RTK is not the same as JAK-STAT. But I believe there is an SH2 in this one, connecting the STAT5 to the receptor, right? Right, guys? Connecting the receptor to the, yes. So that's an example of one. So in this one, I see two. So the STAT5 dimer itself, they have these SH2 domains they connect to, and then the receptor binding to the STAT5 monomer. Great question. Love it. Okay. And the final domain is the SH3 domain. Oh, another question. Sorry. I missed it. Yeah. So I guess I wouldn't say gain, but if you shut down a domain, it's like you're shutting down a connection between two proteins. So, If you shut down, let's say, for example, if you shut down the SH2 domains on AKT, protein kinase 1 and protein kinase 2 will be able to bind there. Because the domain serves a specific function that's important to the protein. So if you shut down a domain, things would not happen as normal. Does that make sense?
Yeah. Is there a game though? Would they be able to separate or is there never a game?
I don't think you'll be asked about a gain of function domain, possibly a loss of function. But the loss of function of a domain means it's cut out, it's canceled, it's not there. Poof, it's gone. That's basically how you should think about that. Any questions? That was an excellent question, by the way. Great question. Okay. Okay. Did I mention SH3? Okay, SH3, the last one, PXXP motif binding site. What is that? Okay, so that is, I don't know, it has like a polyproline binding site for a protein to bind to. I don't really know the deets on that, but just know when you see SH3, it's a PXXP motif. And the X's are variables, so anything could be between like the proline, that's why it's like a polyproline. But just know that SH3, think PXXP motif. Thank God you guys have a cheat sheet to write that down, right? Anyways, so there is an example of this. There's only one example of this actually in your RAS pathway. Where is this? Okay, sorry. One example of this in the RAS pathway, and that SH3 domain is here, and that would connect the adapter protein to the RAS activating protein. Sorry, I'm just checking my notes. But yeah, that is the only SH3, I think, in all the pathways. Any questions? No, okay. So I'm gonna go through the last pathway, and then I can give the floor to the other TAs so they can talk about cell cycle microtubules. Oh, what's going on here? Oh, I put a video. Okay, instead of me explaining it to you, because I thought you guys might be tired of my voice by now, I put a video. So listen to this video, it's important, okay?
Many G protein coupled receptors have a large extracellular ligand binding domain. When an appropriate protein ligand binds to this domain, the receptor undergoes a conformational change that is transmitted to its cytosolic regions, which now activate a trimeric GTP binding protein, or G protein for short. As the name implies, a trimeric G protein is composed of 3 protein subunits called alpha, beta, and gamma. Both the alpha and gamma subunits have covalently attached lipid tails that help anchor the G protein in the plasma membrane. In the absence of a signal, the alpha subunit has a GDP bound, and the G protein is inactive. In some cases, the inactive G protein is associated with the inactive receptor, while in other cases, as shown here, it only binds after the receptor is activated. In either case, an activated receptor induces a conformational change in the alpha subunit, causing the GDP to dissociate. GTP, which is abundant in the cytosol, can now readily bind in place of the GDP. GTP binding causes a further conformational change in the G protein, activating both the alpha subunit and beta gamma complex. In some cases, as shown here, the activated alpha subunit dissociates from the activated beta gamma complex, whereas in other cases, the two activated components stay together. In either case, both of the activated components can now regulate the activity of target proteins in the plasma membrane, as shown here for a GTP-bound alpha subunit. The activated target proteins then relay the signal to other components in the signaling cascade. Eventually, the alpha subunit hydrolyzes its bound GTP to GDP, which inactivates the subunit. This step is often accelerated by the binding of another protein, called a regulator of G protein signaling, or RGS. The inactivated GDP-bound alpha subunit now reforms an inactive G protein with a beta gamma complex, turning off other downstream events. As long as the signaling receptor remains stimulated, it can continue to activate G proteins. Upon prolonged.
Okay, that's it. She explained it better than I could in like 2 minutes. So great stuff. Okay, do people understand how that pathway works, or do you still want me to go through? I'm happy to go through it. Just let me know. Oh,
Many G
no. Okay. Do people want me to still describe it a little bit, or is that a yes? Question?
I know, like in the lecture, she like gave a specific example, like sort of pathway for this. Should we just know that, or should we know?
Just the fundamentals here, folks. Just the basics of how it works. I think. Wait. Yeah, I'm pretty sure. No, I'm sorry. Just don't mind me here. But yeah, just the fundamentals of how it works. And yeah, I'm sorry. I know I spoke for a long time, but yeah, the other hour was supposed to be for cell cycle and microtubules. But do you have a question?
Yeah, is RGS lipid bound?
Is it lipid bound?
Show that.
Not sure. So not sure, but I don't think it'll be on your exam. But also, the video is just an additional tool for extra help. I don't want to conflict the messages between what you got from class and the video, but just focus on your notes you have from this class, how GPCR works. You'll be good. Okay. Another question?
So G alpha and beta and gamma are like receptors, like
Okay, let me-
from each other. So if you like each one with the other would still work, right?
That's a good question.
Because on the
I
problem set,
see.
she said like,
It would depend. And I know that's not the answer people like to hear, but it would depend on what situation is going on there. But basically, I'm going to talk about this a little bit because you guys have the nice summary. And I take it too long. I plan to only take like 15 minutes, but it's okay. But anyways, you have GPCR, and then you have these subunits, like the video said. Ligand will bind to, or signal, excuse me, will bind to GPCR. Big key thing here. When the signal binds to GPCR, causes a conformational shift in GPCR that allows the signal to get like transduced inside the cell. Anyways, the conformational shift occurs in GPCR, and then that will cause G alpha here, which is at this point bound to GTP, to drop GDP. And then because, like the video said, GTP swimming around in the cell and it's more abundant, it'll go in the binding pocket of G alpha and cause G alpha to become active. What does G alpha do when it's active? Dissociates from all of its brothers and sisters and does signaling over here. Okay. And in the video also mentioned as well, these subunits, I think, are, and G alpha as well, they're anchored in this cell membrane. And when things are anchored in the cell membrane, it's just more efficient for signaling, okay? Because if they're floating around in the cytosol, there's a possibility it just might take longer for this downstream signaling effect to happen, which is what Tina was saying earlier with someone else's question about RAS being anchored in the membrane. So it just allows, you know, the signaling that these guys have to do their jobs, they can carry it out more effectively. They're anchored in the cell membrane. Okay. So then you have active G alpha does its thing. I feel like G alpha is like the main character of this pathway. But then beta and gamma, they do their signaling as well. But at some point, G alpha needs to get shut down. So what will it do? RGS will bind to G alpha, and RGS behaves like GAP. Okay, if you remember GAP, binds to a protein and accelerates the protein's intrinsic GTPase activity. So RGS does not hydrolyze GTP and GDP. It'll just help G alpha to do that in a faster way. So that happens. And once G alpha is bound to GDP, I think it links back up with this part and it forms the complex again at every piece itself. Are we good there? Great. Okay, we're gonna jump into the next material. But wait, no, I gotta ask the question. How can we shut this down? You know, I'm always gonna ask this because you guys will be problem solving tomorrow. You're gonna thank me tomorrow. Okay, anyone? Maybe you guys can think about that later. Do you wanna think about it later on, come back to it? No? Okay, go ahead.
I mean, I think one way to probably shut it down is by preventing G alpha from dissociating. That would prevent the activation of G beta and G gamma, and then that would prevent then
Okay, let's say that's a good idea. Yeah. Okay. Yeah. So that's it for pathways. Now I could oh no. Oh, never mind. I thought I was gonna be done talking, but no, okay. Anyways, common misconceptions. I'm only gonna mention one thing on this page. Big thing, okay? The gap. Every time. I've been a TA for 4 semesters now, but every time students do this, and it's just okay. So gap. When you're talking about when gap binds to enzymes and hydrolyzes GTP to GDP. GAP itself does not hydrolyze GTP to GDP because GAP is not an enzyme. It's just a protein that helps regulate other proteins or enzymes. So GAP is not the one that hydrolyzes GTP to GDP. It'll bind to GTPases or enzymes like G alpha that are able to do that itself. It just helps the process go by faster. So that's a big misconception, I'd say. And when talking about pathways, good to just describe proteins and their functions. Okay, and that's it for pathways. Sorry, I know I took long, but we have the other half of the review. Even though it's most of your exam anyways. But yeah, it's fine. Here. Are you gonna say? Okay.
Yeah.
Oh,
I guess I don't
okay,
know.
I'll leave it. Okay, go ahead.
All right, hey guys. Can you guys all hear me? Especially in the back, yeah. All right, so I'm just gonna go through microtubules. So to start, the microtubule spindle, pretty much one of the main things microtubules do in the cell is that they form these spindles, and these are used to separate chromosomes during the M phase. And so you pretty much want to know the 3 different kinds of microtubules, right? Note that they all come from a centrosome at the edges of the cell. And you have the 1 aster microtubules. These grow from the centrosome and attach to the cell cortex, which is pretty much just the inner layer of the plasma membrane, so like the cytoplasm itself. And then you have your kinetochore microtubules. These ones, they grow from the centrosome also, and they connect right here, as their name says, at the kinetochore of chromosomes themselves. And then you have the 3rd interpolar microtubules. These also grow from the centrosome and they attach right down here. And we'll go over later in the review, but these are important, especially for the push-pull mechanism of how chromosomes separate. Moving forward, breaking down microtubules, we'll start at the tubulin, which is the dimer monomer that makes up microtubules and has GTPase activity. So pretty much it just has the ability to hydrolyze GTP to GDP. So breaking it down further, we have 2 subunits, right? We have the alpha on the left side and then the beta on the right. And the important thing to know for these is that they both have areas where GTP can bind, but only the beta, only the plus end has it exposed, right? Because on the alpha end, it's right here in the middle where it can't be accessed. So because of that, pretty much simply that microtubules mainly, especially in the lab or in vitro, will only grow or grow mainly from their plus end or their beta GTP exposed side. Is there any questions on that? No? Okay. So the structure of a microtubule itself. It starts off, like we said, it's made up of tubulin heterodimers, which from the name you can tell are alpha and beta tubulin subunits. They're polar molecules, which doesn't mean anything of their polarity or charge. It means that they're different on each side. So again, there's a positive side, or yeah, a positive side and a negative side, where the positive side is the side that tubulin addition is going to mainly occur. Again, because that's the side of the exposed to GTP. And more, their microtubule is just a hollow tube made up of 13 protofilaments that you can see here. One protofilament is just one column that makes up of the microtubule. So you have 13 of these that wrap around. And especially in vivo or in the cell itself, these are going to originate from centrosomes where the minus end, the end down here, is going to be embedded in the MTOC or the microtubule organizing center. Yep. And you're going to have the positive end extend outward. Any questions on that? No? Okay, so microtubule dynamics in vitro. So again, in vivo, when we say that, we mean inside the cell, and in vitro, we mean in the lab or in the petri dish. So in vivo, first off, there's no growth or shrinkage at the minus end because again, the minus end is going to be embedded right here in the gamma tubulin ring of the MTOC. So you're only going to be able to have it grow from the plus end. And think about how that makes sense in the cell where you're going to have microtubules growing from centrosomes, and they're going to only need to grow from one side to reach the chromosomes or whatever other function they're going to be doing. To contrast that, you have in vitro or in the lab, this is where you just have the pure tubulin or the pure alpha beta dimers, just free flowing in a test tube with GTP. And in this case, you're going to have a more controlled environment where you can see the dynamic instability, which we'll get into later, of how microtubules grow and shrink. But the cons of that is that anything done, again, in the lab is never going to be 100% replicable inside the cell. So if we're good there, we have microtubule enucleation in vivo. So this is the main idea here is that the gamma tubulin ring is formed, as you see here, and act as a base for where the start of the microtubule will grow from. Again, it's going to grow outward from the plus side because the minus end is going to be embedded inside the ring. Okay. So more microtubule dynamics in vitro, again, in the lab. This is going to be done using a spectrophotometer, and it's going to be a time course experiment. So you're going to have a graph here where time is going to be on the x-axis and the rate of absorption or the formation of microtubule is going to be on the y-axis. So pretty much there's three main things to note here. We have three phases. The lag phase right down here, this is going to be the formation of the individual dimers first, the alpha beta subunits, which are going to form together to make oligomers, which are going to then form the column that we talked about, the fluorofilaments. Then you're going to have your elongation phase. This is where the microtubule, all the tubule themselves are formed, so they're going to rapidly grow and shrink until you hit the plateau, which is pretty much where all the free GTP are going to be used up. And because of that, you're going to have no more growth, so hydrolyzation is going to be able to catch up, and you're going to have subunits coming on and off until eventually the MT stops growing altogether. So is that clear for everyone? So going more into first the lag phase, this is going to be the longest step because it takes time for the seeds or for the precursors of microtubules to form. And so the order of that is going to be first, you're going to have, like we said, your free alpha beta tubulin, free flowing. They're going to come together to make the dimers, the alpha beta dimers, and those are going to further come together to make oligomers, protofilaments. 13 of those protofilaments come together to form a sheet, which then close around. And then you're going to be able to go from there to your elongation phase. And the important thing to know, like a popular question she asked, is that what happens the more tubulin you put in? Well, you're going to have a shorter lag phase because you physically have more subunits that are going to be able to randomly come together and combine inside the test tube. And on the opposite of that, how could you extend the lag phase? You just have less tubulin that you put in your test tube. at the start. Okay, all good for there. So we have a question. What if stable aggregates or seeds were provided or already existing inside an in vitro assembly like they do in vivo with the MTOC? Yep. Yeah, exactly. So exactly. So the whole reason that we do have a lag phase is that unlike in the cell, these dimers have to first come together, which again is going to be why the lag phase takes the longest. So yeah. Okay, so then we have the elongation phase. This is when the seeds have been already formed. So then this is going to lead to rapid addition of tubulin binding because again, you have the MT already formed. So it's just going to free GTP is just going to come and add on the positive side. And so again, why primarily to the plus ends and not the minus ends is because those are the ones with the open GTP and have greater affinity. Again, there's going to be some growth on the minus end because this is in vitro. There's no embedded MTC. But again, it's going to be much greater on the plus end because it has that affinity for GTP. And then finally, the plateau. This is going to be when the rate of MT assembly, so the rate of it growing on the plus end is going to equal the rate of MT disassembly. So the rate of it coming apart on the minus end. And this is going to happen because GTP is allowed to catch up, which leads to depolymerization because there's no affinity between the GDP. And also hydrolyzed tubulin cannot further bind. And again, all the tubulin will be used up into its GDP pound. So it's pretty much just that all the GTP is used up or already formed as a part of the microtubule. And again, an important distinction is that this is not that the microtubule is frozen or not growing or shrinking. It's just that the rate of it growing and shrinking is pretty much near equal. So there's no net change in size. So another question, first, in an in vitro assay, what happens after hydrolysis is allowed to completely catch up or it is stimulated? Yeah. Pretty much what happens in the in vitro is that if hydrolysis catches up, that means that there's no free GTP left. So the only thing left for the course is that, because remember again that MTs intrinsically have GTP's activity. So naturally they're going to be converting GTP to the GDP state, which has no affinity. So pretty much when that happens, when all the free GTP is used up, then the MT has no choice but to further hydrolyze and then slowly break off and break apart. And so the second question, how would you cause MT polymerization growing and how would you cause depolymerization shrinking in an in vitro assay?
If you like bombard the assay with ATP back
Yeah.
then you can really increase and make it more favorable for
Mhm.
fertilization. If you either added in or like colchicine.
Mhm.
Yeah, that one which causes rapid depolymerization.
Yeah. So for the recording, the answer was, how would you cause MT polymerization? Simply, you just add more GTP tubulin. So you're adding physical subunits to help with the rate of MT growing. And then for depolymerization, like you said, you would add in something like colchicine, which would cause depolymerization of the MT, which we'll go into more later in the review. So going forward, we have dynamic instability. This pretty much simply says that MTs, individual microtubules themselves, can grow and shrink independently of one another. And that rate is dependent on pretty much a simple whether or not that the rate of GTP hydrolysis, which again is the rate of GTP getting converted into GDP, is that greater or less than the rate of tubulin that's being added. So as you can tell probably that if hydrolysis is greater than the addition of tubulin, then you're going to lead to shrinkage or depolymerization because there's no GTP cap, because there's more tubulin getting turned into the GDP form than the other way around. And vice versa, if you have your hydrolysis less than the tubulin addition, so you're having more tubulin added that can be hydrolyzed, then you're going to have growth or polymerization, and you're going to have that GTP cap on the one end that's going to add to stability. It's all good there. Okay, so drugs that affect microtubules. You have Taxol, colchicine, and vinblastine. So pretty much, like you can see here, the main difference is that Taxol is going to be stabilizing the microtubule itself. And because of that, you're not going to have depolymerization. You're just going to not have any growth. Versus colchicine and vinblastine, they're going to be binding to the subunits themselves, so the alpha beta subunits. And because of that, you're going to prevent further polymerization versus just with Taxol, which prevents destabilization. All good there. So another question, you add colchicine in your in vitro assay at time 0, so at the very start. What results would you expect to see? Yep. Yep, exactly. Since we're starting it at time 0, this is going to be before the lag phase even gets a chance to happen. So the colchicine would prevent pretty much any of that from happening at all. Are you? Okay. Okay, so now MAPS, or microtubule associated proteins and motor proteins. So pretty much for MAPS, the main important one is just tau, which we went over in class. It's highly expressed in the CNS, or the central nervous system, mainly in the axon of neurons. So it's very important for a signal transduction or neuron transduction, I have to say. And it pretty much binds to microtubules and stabilizes them to aid with signal transduction, like I said. And pretty much all you need to know for tau is that in its dysfunctional state, it can lead to disease such as dementia, Alzheimer's. And this happens pretty much because it gets hyperphosphorylated and then can no longer bind to the microtubule, which leads to it aggregating, entangling, like you see here. So good there. And then we have motor proteins. And these proteins, they hydrolyze ATP to move along microtubules, like you would see cars that use roads to transport cargo or just go around in the cell. And pretty much you need to know is that there's 2 main kinds of motor proteins, and the distinction between them is the direction which they walk. You have kinesins, which are plus end directed, so they walk towards the plus end of the microtubule, like you see here. All of them except for the kinesin 14 family, which walks towards the minus end. And they're more diverse, so they have different family of kinesins, and they perform different functions, like you know that like that table we went over in class. And then you have dyneins, and conversely, they walk towards the minus end, and they're common in structures like flagella or cilia. Okay, so the opposing forces of motor proteins. So like we were talking about before, back in spindle formation, it requires an array of opposing forces. Again, you know, like kinesins, they go towards the positive side, dyneins that are pulling towards the negative side. And this opposing forces creates a tension that holds the chromosome in place before it gets pulled apart. An example of this is AG5, the kinesin that binds to interpolar microtubules that overlap right here. Since you can see that it's plus n directed on both sides, you can see that they walk in opposite directions, and this pretty much goes to push the spindle poles apart. and it can be inhibited by monstro, which pretty much collapses the entire spindle. So you see here that this would be the egg 5, and they're attached on both on the top side and the bottom side, walking opposite directions again, like we said. So another question, how does kinesin 5 or egg 5 push the spindle poles apart? Yeah. Oh yeah.
With alignment of microtubules, like in terms of like maintaining the overlap with
Mhm.
like is that just to maintain the overlap of the interval?
So you're asking why they have to overlap?
Yeah, like what part
Yeah.
is actually overlap?
Oh, you mean oh, the mic tube itself? I would say they overlap, but only because I can go back. So you're talking about the overlapping here.
Okay, so that's the overlap.
Yeah,
I
yeah.
just didn't know what
Oh,
overlap
yeah,
was.
yeah. So yeah. Yeah, so that's when they say over.
Circles.
Yeah, so the circles here, they represent the egg 5, and that's where the spindles like overlap right there, and then they're pushed apart. Okay. So yeah, so pretty much like we were talking earlier for this question, the way they get pulled apart is that egg 5 is going to be walking towards the plus end on both the top and the bottom, and that's going to push apart the spindle poles.
Okay, since you guys have access to the slides, you can read on the different types of kinesins, but we're gonna jump right in with cell cycle. Thank you, appreciate it.
So there's like half an hour-ish left. I'm gonna try not like speed through necessarily, but more so just focus on the parts that are a little more extra important that you'll probably see on the exam. All right. Huh? Take it as you will. Okay, so the cell cycle. Oh, no, you're good. There you go. So just a little bit of a general overview. This shows you as a whole what the cell cycle is. Something to really emphasize is that even though it shows that G0, it looks like it's the start. It doesn't necessarily always start at G0. It can go into G0 at any point within G1, up until like middle of G1. But cell cycle itself does start with G1 rather than G0. Okay, so a little bit about cyclin-dependent kinases. So they are the master cell cycle regulators. So if in any case throughout the entire cell cycle, you wanted to kind of shut down or stop the cell cycle, a lot of the ways that scientists will try to stop the cell cycle is by targeting specific CDKs. If you don't have the CDK, then you can't continue on with the cell cycle. So a lot of like cancer treatments, they try to target CDKs because that'll stop the cell cycle. They are kinases. So I think that was one of the questions, like an iClicker question in class, but the cyclins are not the ones phosphorylating or doing anything enzymatic. The CDK itself is the kinase part, not the cyclins. So some people get that confused. And so just to clarify, CDKs can be regulated by a few different ways. They can be regulated by either cyclin levels, phosphorylation or dephosphorylation. Again, to clarify, sometimes students think that phosphorylation always means activation, but in the case of WEE1, like we've learned, that is also phosphorylating, but that's definitely not activating. It's actually inhibiting CDK. So WEE1 is an inhibitory inhibitory kinase that's putting on an inhibitory phosphate. And then CDKIs mean CDK inhibitors. So there are certain types of CDK inhibitors, such as P53, which I'm going to go into a little bit more in detail as well. But it's really important also that just because you have cyclin and the CDK bound together, that doesn't mean it's active yet. It also requires the We1 phosphate to be off and for the activating phosphate to be on. So they need both the activating phosphorylation and the cyclin, as well as for the We1 phosphate to be off in order for CDK as a whole to be activated. Right. So a little bit more about CDKs. They target different substrates or proteins to activate or deactivate depending on what cell cycle phase they are in. So each cycle of the cell cycle has a different cyclin. So then that means CDK binds to different cyclins within each phase. You don't need to know very specifically what each one does, just that in general, you do have different cyclins and different CDKs binding within each phase of the cell cycle. Yep, and then just to clarify, or not clarify, to emphasize, CDK can't work as a kinase unless it's bound to cyclin. Oh, yeah, that's right here. Sorry, I lost my train of thought. And then to go on to cyclins. So while CDK is regulating a few different ways, cyclin is only regulated by production or degradation or destruction. So this shows a little graph of which type of cyclin is created during each phase. So like I said, each phase kind of has their own specific cyclin. So they're kind of increasing and decreasing within each phase. So essentially, cyclin is regulated by its own concentration amount. And so cells produce the cyclin that they need in order to complete the specific cell cycle phase that they're in. For example, in N phase, CDK needs cyclin B. During S phase, CDK needs cyclin A, so on and so forth. Once that cyclin isn't needed, usually it is degraded so that the cycle can continue on without being confused about which cycle it is in. And this part is really important. So again, you need cyclin in order to move on with the cell cycle. If cyclin is not there, most likely you probably won't be able to continue cell division. Does that point make sense? All right, awesome. But yeah, does anyone want to explain really quick why you can't continue on?
So basically when cyclin binds, it changes the conformation of CDK, so it moves away this like T-loop structure, and it opens it up for phosphorylation, I believe, by CAC, CDK activated kinase, and then it can go on phosphorylated targets. Yeah, exactly. So again, because of everything that you just said, if cyclin isn't there, CDK can't do its job, and then the cell cycle can't continue or move forward, and you most likely wouldn't have cell division. So the CDK cyclin complex together, this is exactly what you just explained, the activation loop. It's inside the CDK and it's not available to bind to anything until the CDK is bound to the cyclin. So cyclin binds to CDK, it undergoes a conformational change. That activation loop is now exposed. And so the CDK can now phosphorylate other proteins because that activation loop is exposed. And you can also have CDKIs or CDK inhibitors. And what they do essentially is they block the CDK from phosphorylating the target proteins. So they block CDK from binding to other substrates so that the cell cycle pauses or is braked so that it can't continue on forward. And an example of CDK inhibitors or cell cycle arrest would be P53. So for example, if your DNA has a mutation in G1, one example that Laura really likes to go through is P53. So it's really important to understand what P53 does and like how it goes about its entire process. So P53 is a transcription factor. It is also a tumor suppressor. So because P53 is part of cell cycle arrest, that means it's trying to stop the cell cycle. And because it's trying to stop the cell cycle, it's like a tumor suppressor.
because if you don't have a lot of cell division, you probably won't have, or you won't get cancerous cells. So it's the opposite of an proto-oncogene, which is like a cancer cell. So that's really important to note. P53, it's a tumor suppressor.
Oh, Tina, you said that like 3 times.
Yeah.
It must be important.
Okay. And then it also activates, the way that it acts as a tumor suppressor is that it activates the transcription of P21. So P53 is the transcription factor, but what's actually doing the inhibiting of CDK is P21. And so again, once you inhibit CDK, the cell cycle is put on brakes. That allows the cell to try and, figure out if it can fix the damage or if the damage they deem is irreversible. P53 can also signal for apoptosis, either like ubiquitinating it or bring it to the proteasome. Essentially, if the cell cycle thinks that whatever damage done was too far beyond repair, it definitely doesn't want that cell to be divided, so it's going to try and get rid of it. So just a little bit of a review question. Why is P53 a tumor suppressor? Oh, yeah, go ahead.
Because if the cell is mutated, the cell from continuing to mutation, which if it's allowed to,
Yeah, exactly. And then just to add a little bit to it, P53 then transcribes P21, and that like actually inhibits the CDK. And then everything after that was awesome. So it stops the CDK, and then that's what puts a brake on the cell cycle. And so what would happen if P53 is no longer available? Yep, it'd probably become cancerous, the cell. Awesome. Oh, yeah. Oh, like how does P53 get activated in the first place? You don't need to know. And since it is a transcription factor, I'm assuming it's a lot of like backstream or upstream scenarios that caused it to be,
Probably like cell signaling.
yeah, to like have created it in the first place. Yeah, exactly. And then kind of like the role that P53 plays as a whole.
That is more important. What's your name?
can you go over again why it's a tumor suppressor?
So P53 is a tumor suppressor because it goes to transcribe P21. And what P21 does is that it inhibits CDK. So P21 can bind to CDK, and then that inhibits CDK's ability to continue to phosphorylate whatever it needs to phosphorylate. And so since CDK is the master cell regulator, if CDK can't do its job, then the cell cycle can't continue on. So do you kind of see like that pattern of P53 creates P21, P21 inhibits CDK, CDK is not working, so the cell cycle can't go on. And so if the cell cycle can't go on, there's no cell division. So that suppresses a tumor. So a tumor is like if you have a lot of cell division, but because P53 is stopping cell division, it's suppressing the tumor. Does that make sense? Okay. Any other questions? All right. So going into G1 phase and the pathways within G1, or not the pathways, the mechanisms. So G1 is essentially just looking for if the cell is big enough, if there's enough nutrients, is there DNA damage again? So if there is, that's the whole like cell cycle arrest, P53. In G1 CDK, it is bound to cyclin at that point, but it's not active yet because it has to have like the active phosphate and then get rid of the inactive phosphate or the inhibiting phosphate. So this is a little question that I feel like sometimes a lot of students don't really think about, but is equally as important. So what does the cell need to start thinking about starting cell division? So there's something that signals the cell to start thinking that, oh, it should go into cell division. Does anyone know? So it's a growth factor. So if you remember from Michelle, the RTK RAS MAPK pathway, that's the growth factor signaling that a lot of cells use in order to signal cell division. So the cell has to have the growth factor signaling in order to start thinking about cell division. And so as a result, once it has that growth factor, the cell will create the G1 cyclin. So growth factor from the RTK RAS MAPK pathway signals the cell to want to start dividing, and then it'll create the G1 cyclin. Okay, so the G1 phase is also where the pre-RC is formed, and it's really important that the pre-RC is formed when CDK activity is low, because once CDK activity is high, it's going to start bringing in a lot of the pre-IC proteins, and the pre-RC always has to form before the pre-IC. Does that make sense so far? Okay. This goes over like the details of what the pre-RC contains and what it has. You don't need to know very specifically like every single part of the pre-RC, but for your own studying sake, if you want to go back to this slide and look at it, you definitely can. Okay, so then P53, if the DNA is damaged, that's what we just went over before. So then we're going to keep moving on to the restriction checkpoint. So this is the point of no return, as Laura likes to call it. It's when the cell commits to the cell cycle. And when everything is in check, everything looks good to the cell, the G1 cyclin CDK is activated and phosphorylates RB. It phosphorylates RB, not E2F. RB is part or is holding onto E2F. So once RB is phosphorylated, it's gonna let go of E2F. So in this picture, RB and E2F are together. CDK cyclin comes in to phosphorylate RB, and then RB lets go of E2F so that E2F can go into the nucleus and transcribe all of the proteins needed for S phase. And then that's why it's called like the point of no return, because you're having all these S phase proteins. So the cell cycle has what it needs in order to continue on to the S phase. So it's going to continue on into the S phase.
So is pre-RC, is lots of like pre-RC information
I would say that the point of no return has more to do with the RBE2F mechanism. Yes, the pre-RC is like a little separate part of it. That's where like you need the origin firing to occur. But in regards to how the cell makes it past the restriction checkpoint, it definitely has more to do with RB and E2F.
Like how does cyclin know to activate it? How does like track
Yeah, so you don't, not specifically, you don't need to know, but a lot of times I've asked that before too, and Laura's answer is usually that the cell has like a feeling. The cell just kind of knows, and it has like this feeling of when to start. Sometimes it is signals, sometimes it's that feeling that she talks about. Unfortunately, I don't have that specific answer. I'm sorry. Okay. So any question about how to make it past restriction checkpoint? Yeah. Oh yeah, usually it is before the restriction checkpoint. I've asked Professor Francis as well. The restriction checkpoint and like pre-RC, pre-IC are a little bit like wonky. They're not necessarily happening at the same time, but they're not entirely separate things either. You don't need to know how like the timing of that though. Yeah, so pre-RC, I would know the main points that pre-RC has to happen when CDK is low. So that's definitely beginning of G1. And then restriction checkpoint is sometime closer to the end when there is the active CDK around to go and phosphorylate the RB. Yeah. Yes, RB, let's go of E2F, and then that's when the restriction checkpoint continues on. Okay. So these are just super quick, like true and false questions. If P53 is suppressed or damaged, what happens to the cell? Yeah, it probably becomes cancerous. True or false, in response to DNA damage, cells should increase concentrations of CDK inhibitors. Yes, because if you want to stop CDK, you want to increase CDK inhibitors. So for example, P21. Number 3, true or false, in response to DNA damage, E2F is active. Yes, exactly. Number 4, could E2F be a cause of cancer? Can a cell divide if E2F does not function? Sorry, one person? E2F is the one that causes the cell to make it past the restriction point.
Yeah.
So would E2F in its own, like whatever E2F does, could that be a cause of cancer? Yeah. Yeah, so if you had like gain of function in E2F, or if you had overactivation of E2F, anything like that, that could cause a cancerous cell because the cell cycle is going to keep going. And so if you don't have E2F, there would be no cell division because it can't make it past the restriction point. Nice. True or false, pre-RCs can form at the end of G1 after CDK is fully activated. False. Yay. Okay, cool. Moving along. S phase. Okay. So S phase is when the cell is also looking for DNA damage. So because of that, P53 can also happen within S phase. It's essentially the same mechanism, so I won't go too deep into that. S phase CDKs inhibit new pre-RCs from forming. So that's the whole idea that pre-RCs can only form in the beginning, because once you make it past the restriction point, your CDKs are active. And so when CDKs are active, they're going to phosphorylate ORC. That's going to prevent the association between CDT1 and CDC6. So ORC will not be able to bind. It's going to phosphorylate CDC6, and that's going to lead to MCM not being able to be recruited. MCM is the helicase complex. And then it can also phosphorylate MCM to get exported out of the nucleus. So pre-RCs cannot form in S phase, and you don't want it to form in S phase because you only want to replicate DNA once to try and reduce the amount of damage that could happen. Does that make sense? So like the key takeaway from this point would be that after restriction point and into S phase, the CDK is going to go and phosphorylate all these things to make sure that the pre-RC is not forming. Okay, I saw a thumbs up. Nice. Okay. Okay. Thanks. So in the S phase, that's also when cohesin rings are bound. So you have the activation of the origins of replication so that your DNA polymerase can bind and start replicating. And then cohesin rings form around those replicated DNA. It's important for cohesin rings to form during the S phase, because if they come in to bind later on, you might have the problem of the chromosomes by that point, they'd be like somewhere in the cell where they can't easily find each other. So then that could be a problem, because once the cells do divide, you might have like a different amount of chromosomes within each cell, something like that. any questions about S phase?
Is PRI, what's like the thing after P?
Pre-IC?
Oh, pre-IC.
Mhm.
Is that part of S phase or G?
Pre-IC is part of the end of G1 after the restriction point. Yeah. Okay. So then G2 phase, we're almost done. G2 phase is when the cell is growing again. So it's prepping for chromosome segregation. This growth phase is really important because it tells you whether or not the cell is going to be too big, too small. And then that has a lot to do with the We1. So for example, if We1 is on for an extra long time and it can't be taken off, it's going to be stuck in G2 phase for a longer period of time. So then that means your cell is probably going to be huge or at least larger than usual because it's stuck in that G2 phase longer. If We1 was taken off earlier, then your cell is probably going to be smaller because it left G2 earlier. Did that make sense? All right. Okay. This is essentially what I said before. So as a whole in G2, so you have your CDK, you have your cyclin complex, you have an inhibitory phosphate put on by We1, an activating phosphate put on by CAC. And so right now it is still as a whole inactive. In order for it to become active, oops, it does not show, does it show? Yeah, so in order for it to become active, CDC25 has to come in and it's going to take off that inhibitory phosphate so that the whole MCDK is active. And it is a positive feedback loop. So the more active MCDK you have, it's going to go activate more CDC25s, and then that's going to increase the amount of active MCDKs. So it's this positive feedback loop where an increasing amount of active MCDK is created. Okay, so what if there was a gain of function mutation in CDC25? In CDC25, so CDC25 is the one that takes off the We1 phosphate. Oh yes, sorry. And then would the cell be larger or smaller? Yeah. Number two, what if We1 had loss of mutation? Mm-hmm. Yeah, exactly. And then same thing, the cell would be smaller. And then if We1 had, which one? Oh, okay, I can say that then. Okay, yeah. Just to go back to like the first two questions. So they would be smaller also because it's going to leave G2 phase earlier. So if it's leaving G2 earlier, it doesn't have as long of a time to grow. All right. And if We1 had a gain of function, so that's the opposite of number two, the cell would probably become larger because it's staying in G2 longer. Yes. All right. Awesome. So M phase, that's spindle assembly and chromosome segregation. This is just a generic overview of M phase, the mitotic phase itself. So it's PMAT, you have the prophase, metaphase, anaphase, and then telophase / cytokinesis. This is just kind of like for your own sake of review. Okay. So in prophase, you have active MCDKs. So you have to have MCDKs in the beginning of prophase because they are what causes the nuclear envelope to break down. They are what causes the chromosomes to condense. And then they are also what causes spindle assemblies or the spindles to form. So it's like those main three things. In metaphase, you have the sister chromatids. So they're attached to each other by the kinetic core. It doesn't show it here, but kinetic core is usually in the middle. It attaches the two chromosomes together. They are secured together by the cohesin rings that we talked about from S phase, and then they're lined up along the metaphase plate. From metaphase going into anaphase. Yeah, from metaphase going into anaphase, this is the spindle assembly checkpoint, because at that phase, the cell has to make sure that all the sister chromatids are aligned in the middle of the cell. So this is kind of like a sensation that the cell feels. Once it feels like an equal tugging on both sides, that indicates that all the sister chromatids are in the middle. And so then that equal tension allows CDC20 to be released. And then that's what causes metaphase to go into anaphase. For this exam, you don't need to know very specifics about all of the different phases within mitosis. So as long as you have like a general understanding of what happens when, that's good. Yeah. Okay. What time is it? Oh, 9:04. Okay. So the kinetic core, again, this is where this is where the two microtubules are connected together. Oh, the microtubules connect to the kinetic cores at the center of the replicated chromosomes. That's all you need to know, that the kinetic core is at the middle. And should we go over the push and pull one? No.
Do people want us to go over the push and pull?
Yes, please. Is it on the exam?
That's possible.
Oh.
From what I understand, though, folks, the exam hasn't been finalized yet, but there's a planning.
Okay. So
And then the plan.
I think a
But
lot
I
of,
think because people had questions about this, we're gonna go through it.
yeah.
But please listen to me. Thank
So
you.
a lot of students, I think you guys have like a hard time understanding like which way the movement is, right? Okay, so think of it as in this picture, the arrows show which way the spindle poles are being pushed or pulled. So for example, this one, so these are egg 5, so kinetic kinesin 5s. It's showing that the arrows are moving outward. So that means the spindle poles are getting pushed outward. And this is because in egg 5's case, they are plus end directed. So they're going to try to walk towards the plus end. And in turn, you can think of it as like you're dragging your foot or dragging your hand. So if you're going towards the plus end, you're like scooping backwards. So the spindle poles are moving opposite direction. Yeah.
Is this like
Anaphase B, that would, yeah, because that would be the spindle poles.
So she's just going through it because it was a concept a lot of people had a difficult time understanding the discussion and everything. Can't guarantee or not guarantee will not be there, but just try to understand what she's saying. Okay?
Yeah.
Thank
So
you.
just the overall understanding is that whichever way the dynein or kinesin is moving, your spindle pole direction is usually in the opposite direction. So for example, this one, it's a minus end directed dynein. So it's trying to move towards the minus end. So because it's moving towards the minus end, it's dragging it like backwards. So it's going to be pulling the spindle pole outward. Does that make sense? Helps a little bit? Yeah. This one? This one, yep. So that's like the push and pull part. So if the egg 5s are causing the spindle poles to move outward, this dynein is a minus end directed. So it's going to be walking towards the minus end this way. and then that means the spindle pole is going to be moving the other way. Yeah.
I just thought of something that like thinking it as a treadmill, like you're walking about the treadmill.
Yeah, exactly. Yeah. So whichever way you're walking, the spindle poles are moving in the opposite direction.
Love the analogy. Making over there, see you in the making.
Mhm.
Just go to that.
Yeah.
I'll do like the close up thing.
Okay, because I think, oh, okay, there's only like a couple more slides. Yeah.
So we're knowing like which direction to go, should we know like where the plus and the question that is at the organizing center sound organizing.
So plus end is at the end of the microtubules. Oh, that doesn't help. So minus end is the end that's like in the spindle pole.
Yeah.
And then the plus end would be like the more free side. Yeah, generally you just need to know one is minus, the other is plus. Yeah. Any other questions?
The videos say where the minus, like for the microtubules,
Mhm.
the videos mention what's connected in the notes as well.
Yeah, Michelle was just saying that like in the videos too that Laura provides, and in her lectures, like she talks about which side is the minus and plus.
From the microtubule portion. Yeah, also in the videos and the reviews.
Yeah.
But go ahead. Tina.
All right.
You're doing great.
So then spindle assembly checkpoint. CDC 20 will bind to the APC and activate APC. APC is a ubiquitin ligase. So that means that it's an enzyme that will target proteins with ubiquitin. And once you target something with ubiquitin, it's going to be sent to probably the proteasome. And that's where whatever it got targeted is going to get degraded. So securin is bound to separase. And once APC is bound to CDC 20, so from this first step, they can go and target securin. And because APC is a ubiquitin ligase, it can cause securin to be degraded. Securin, again, was bound to separase. So if securin is no longer bound to separase, separase can go and cut off the cohesins. And with the cohesins no longer binding the chromosomes together, they are free to leave metaphase and split apart for anaphase. Did that make sense? All right. So just a little review about the different types of anaphase. There's anaphase A and anaphase B. This is, in my opinion, the most simple way to think about it. Anaphase A is anything that has to do with the chromosomes being pulled towards the poles. So anything from the middle going out, specifically having to do with the chromosomes. Anaphase B is anything that can cause the spindle poles to be pushed further out. So whether that is interpolar microtubules like the egg 5s that we were talking about, those can push the spindle poles outward. Dyneins on the aster microtubule. So if you have dyneins on this, they're going to be walking minus n, and then that'll cause the spindle poles to be moving in the opposite direction. So anaphase A, anything to do with the chromosome specifically. Anaphase B, anything that's going to cause the spindle poles to be moving outward. That makes sense? Any questions? Cool.
Oh, we included this diagram here. Throwback to 3 years ago. And I said, what if you want? You took Professor Phillips, you know this diagram. So there you go.
Yeah.
Threw it in there. Summarized it nicely.
All right. And then telophase and cytokinesis. Sister chromatids have finally reached the opposite poles. Your spindles are breaking down. Nuclear envelope forms around DNA. And the way that this is all happening is because you have the APC. So APC also targets M-cyclin CDK. So if M-cyclin CDK is degraded, if you remember, M-cyclin CDK was what originally caused nuclear envelope to break down, chromosomes to condense, and then spindles to form. So now that you get rid of the M-cyclin CDKs, the nuclear envelope can form back up, the spindles can deform, and then the chromosomes can decondense. And then, yeah. So these are just questions to think about. All of the answers are in the speaker notes that we're gonna send out. Yeah.
Hang on, hold on. Okay, so give me one second here. Okay, YouTubers. So this is the end of the review session. All the materials for the exam are listed here. All our TA review videos on YouTube and the slideshow links because we're nice like that. And like and subscribe to my channel, please. And that's a wrap with the review session. Thank you and good.